Good afternoon, everyone. Welcome to the session 3 of Asian Solanaceous and Cucurbits Roundtable. So we will start very soon. So my name is Sheshshayee, Executive Director of APSA. We would like to welcome everyone again to this webinar. So I just would like to give a brief instruction on how to use Q&A function, because this is the first time that we just add more function to the Q&A box. So in here, if you would like to put your questions, you just go to Q&A box, all right? And then once someone put the question, you are able to view that. And if the question that has been asked before, it is the same one that you wish to ask, you can click like. So once the question got the most like, it would move up so that it draw an attention to our speaker to give an answer to that question. All right, so you can try to see now in the Q&A box, our secretariat will just put the test question. Xiaofeng, are you able to put the test question? Oh, someone else put that. Yes. So you see, yes, now you can see the like function, right? So if you can keep viewing the questions during the presentation given by our speaker. And then after each speaker finished their presentation, we have 20 minutes time for each speaker to go to each question. So basically, they will just, the speaker will select the top 5 questions based on the time available. But I believe that 5 questions would be good to go during the session of 20 minutes. And then the rest of the questions we will share to our speaker after the webinar. And then once they answer all the questions, we will share in the summary report. And I think for the rest of the instruction, you should be able to get familiar by now. With that, I just would like to welcome our chair of our R&D advisory group, Dr. Simon, to give a welcome remark. Dr. Simon, please.
Yes, welcome also from my side, and also my colleagues in the R&D advisory group of APSA. We just had a short preparatory session, and Ivo, the first speaker, Dr. Ivo Rieu, had not shown up, but I see him now in the participant list, so I think everything is ready. So we have two very good speakers, both specialists in crop physiology. First speaker, Professor Ivo Rieu of the University of Nijmegen in the Netherlands, not completely unknown to many of you, because he's quite well known in the Solanaceous community, and he has also talked about his research in ASRT. The second speaker is from University of Agricultural Sciences in Bangalore, Professor Sheshshayee M, professor in crop physiology. But let's first go to the first speaker, and then continue with the second speaker during the second part. I will unfortunately not be able to sit out the full session. I have another meeting coming up later, but I'll introduce the second speaker halfway. Enjoy the meeting. Ivo?
Yes. Can I share my screen? Yes, I can. So do you see my screen?
It's perfect.
Yes, perfect.
Yeah. Okay. Good. Then, well, thank you to APSA and Sreeman for inviting me to talk here. And indeed, I have been I've been presenting my work to you a couple of years ago in I think it was in Bangkok, and it's a pleasure to be back again. So my talk is about reproductive heat tolerance of tomato. Overall project name heat yield in this case, yeah, so I will present you what I've been working on for the last couple of years with my team. So very quickly through this, of course, we all have heard about global warming, and global warming is normally seen as and higher average temperature that we have on our whole globe. Well, although this might give some problems to plants, this average temperature, for example, in phenology, in flowering time, it's not really a stress for most plants. So, but what is a stress and what is also associated with global warming is that we have these more extreme weather events. And in this case, I would like to draw your attention to heat waves. So here's a prediction between 2001 and 2057. It's for the US, but I'm sure you can apply this to many regions around the world. And you can see that predicted is there is a higher frequency of heat waves, but also the duration of each heat wave will be longer and the intensity, so the absolute temperature will also be more extreme. So we're really facing a problem here. And why are we facing the problem as a society? Because plants, well, all organisms, but also plants, are very sensitive to changes in temperature. And, well, this is a very conceptual picture where you see what kind of processes are affected by temperature. And well, maybe we should actually see it as almost a physical biochemical problem, that with higher temperature, molecules and atoms, they get higher energy levels, they start to move. And well, that means at one hand, for example, many reaction rates are faster, but on the other hand, also many molecules are affected in their structure. So after a while, for example, enzymes start to become less effective. And well, this happens with all kinds of biochemical reactions in the plant. So altogether, that gives this blue growth curve that you have somewhere. There's an optimal where everything is well in homeostasis, and you get the best growth rate. But if the temperature goes higher, the growth rate drops. Well, having said this, if we look at different processes in plants, and here it's separated a reproductive response to temperature and a vegetative response to temperature. And I think this holds for many plants, at least that I have looked at in literature, is that the vegetative phase is more tolerant to higher temperature than the reproductive phase. Okay, so here's the response curve, and here there's, in this case for soybean, but as I said, it holds for many species, also many crop species. The vegetative optimum range is at a higher temperature than the reproductive optimum range. Well, and that is, of course, exactly counter what is happening in nature, because most of, and also on our agricultural fields, because mostly the reproductive phase is in the warm period of the year. So what happens to this reproduction if it then encounters this high temperature in the form of heat waves, and you see it here for different species, monocots, and here on the right is probably more interesting to you and more familiar to you as well, is that tomato becomes heat sterile. That's what we call it, heat sterility. And in this case, I think there's only one fruit here formed with seeds, and probably all the other ones are just parthenocarpic remnants. Okay, so this is a problem for agriculture, and well, usually I think with many problems, breeders can deal with those problems themselves very aptly. But this has been more difficult for heat tolerance. So I've been talking to many breeders over the years, and I think there's 3 problems. Well, let's first look at the first 2. They're related actually to the third one, is that breeders usually they don't have the time and the means to really focus on the reproductive process itself, how well that happens. Instead, they look at the performance of their crop by looking at yields. And we all know that yield is a difficult trait because it integrates the whole plant physiology. And that is what happens. There might be other stresses in your plants or other conditions that affect your yield at the end. You can even have diseases, something like this. And that's all integrated into this yield. So dissecting out the reproductive heat tolerance in such a field is difficult. And then even if you have a nice high temperature period, well, at least for dissecting, it's nice to have it, but of course you probably don't like it for your yield. But if that happens in a season, it still doesn't always reflect very clearly in the yield, because the yield, of course, integrates temperature over the whole growing season. So it might be only a part, and it can be a few percentage, up to actually more than that, but yeah, it might be difficult to recognize this. And I've been talking to breeders that really have trouble even, let's say, ranking their own germplasm or their own varieties for heat tolerance because of exactly these problems that they look at in a year and actually there's no heat wave that year, so that then they don't get the right information. Well, and thirdly, and it's, as I said, it's actually related to the former two, is that, well, it is actually quite difficult to look at reproductive heat tolerance separated from all the rest, because it's a difficult process, and we don't have to go through this figure in detail, but here we start with a flower, and of course you all know we have a female development, we have male development, and we end here with the spores and the gametes, and they form a zygote, which goes back through the vegetative phase to produce at the end the flower. So different processes in this scheme can be affected by heat. There might be effects on the female side, it might be effects on the male side. Actually, the pollination itself can be affected, maybe early seeds. So it is quite difficult to look at that specifically for a breeder. So what we set out with my team is to, well, we had this goal. Our goal is to improve reproductive heat tolerance, because it is something which needs additional attention. So to come to this goal, we asked ourselves two questions. And the first question is, what is actually going wrong with fertility if you have heat stress, if you have these heat waves? And the second question is, can we identify or generate tolerance? And that could be by genetic means, but it could also be by management things, by management means, for example, treatment with certain chemical. Well, you can, of course, label these things quite easily, and you would maybe say, well, the first question is mainly going to generate fundamental insights about the mechanism, and interesting, that will be published in the scientific publications, and the second one is going to deliver the strategies and the applications that we need to really solve this problem. Well, and then actually, if you look at it like this, you might even say, as a breeder, well, why do we need this top question if we want to have the solution? But if you think about it a little bit better, I'm sure you agree with me that these two questions heavily interrelate, because if we know what is going wrong with fertility, that might give us strategies to actually generate tolerance by a genetic means or by treatment means. The other way around, if we know that there is certain tolerance, either genetically or by treatment, we can actually use that to dissect what's happening. Okay, so my statement is that, point of view is that these two things are heavily interrelated, and we actually need both to answer both questions to be able to answer any. Okay, so if we want to get answers to this question, we need some facilities. So in Nijmegen, as Simon just announced, is my university, is in Nijmegen in the Netherlands, and we have a few facilities. So we have a greenhouse, and inside this greenhouse we even have growth cabinets and climate chambers that we can use to apply certain environmental conditions. And in this case, we apply something which we call long-term mild heat. So that's the LTMH that you see here. Change my pointer a little bit, that's maybe easier. So here the long-term mild heat, and we compare to control treatment where we have a nice temperature for tomato for growth and reproduction. And here it is, both of the day and the night temperature are increased, and we call it long-term mild heat. And that indeed leads to fertility problems. Well, I'll come back later to you whether this LTMH, whether using long-term mild heat is actually a good choice if you want to target heat waves in a field situation, because heat waves are, of course, not a block temperature of exactly 32 during the whole day and 26 during the whole night. There's a profile in the field. So I'll come back to that, whether actually this is a good choice for studying heat waves. And I can come back to that because more lately we've not only been doing experiments in the controlled environments, but also took our plants to the fields in Spain and Italy in this case, and soon we will also be doing experiments in Turkey. All places where you have heat waves during the flowering stage. Okay, so I also already explained to you that that reproductive is a difficult process, or not difficult, it's a complex process, many things happening at the same time to get a to get good fertility. And well, instead of targeting everything at the same time, we said, well, we need to focus a little bit and tackle the different problems one by one, because otherwise we won't be able to find anything. So here's an example of what we did. Here we looked at several traits: pollen number, pollen viability, and style protrusion under high temperature. And we said, how well does that correlate in this case to fruit sets? So those are non-parthenocarpic fruits, or even the number of seeds. It's very comparable. And if we correlate, let's say in the big data sets that we have, if we pull all those data sets together, we get such a picture where, for example, pollen number has a positive effect. It's not very strong. It's probably because normally there's a huge overkill, let's say, in the number of pollen that a tomato flower produces. So there might be 50 or 100,000 pollen in a flower, whereas there's only place for 2,000 pollen on the stigma, and there's only place for 200 seeds in the fruit. So you understand the pollen number, even if it drops dramatically, you might still have enough pollen to get good seed set and good fertility. Something different with pollen viability. So consistently, we see that this trait, pollen viability, really influences seed set and fruit set. And here's a negative factor, style protrusion. That is something that in some genotypes still happens if you put them in high temperatures. Styles tends to be exposed to grow a little bit higher than the anthers, and that prevents or hampers pollination. And you can see indeed that you get negative correlation. Well, we chose to focus first on pollen viability. I will say something about, or you will see something about pollen number and style protrusion, but pollen viability, and that's also because, for example, style protrusion has been already solved more or less in the most varieties that have non-protruding styles, even under high temperature. So this is what it looks like. If you look under the microscope, this is pollen from control temperature, and this is pollen from this long-term mild heat. And here you see that most of them are dead. So that is the pollen viability trait. Well, I've not explicitly said it to you, but you've seen it by now that most of my research is in tomatoes. So actually my whole talk will be about tomatoes. So we've also been doing some experiments in brassica and in Phaseolus and common bean, but that's not part of this presentation, also not a major part of my research yet. Okay, so we also need methods to phenotype the pollen. So here we have the classical methods of a germination assay or a staining assay, the many viability staining assays described in literature, but those are all very low throughput. So instead we are using this impedance flow cytometer, and I think many bigger breeding companies also have possession of these types of machines, because it can very quickly discern live pollen here, which would be on the right side of the graph, and here in this sample, actually this live pollen section is empty, and actually all pollen are dead in this case. So a very essential machine for us to quickly phenotype our plants. Good. So having said all this, I would like to answer the two questions with you, at least to go through them. So the first question was what is going wrong with fertility? And in this case, we focused this a little bit further to say what's going wrong with male fertility under high temperature stress, under heat waves. So we wanted to first answer some very basic, what we thought were basic questions. So for example, where is the damage done? Because the whole plant is usually exposed to high temperature. But yeah, we see a phenotype in the flower. So is the effect of the high temperature, is that specifically on that flower that shows the phenotype, or is it indirectly, is it actually an effect on the whole plant, which then translates to problems with pollen development? So we did this very simple experiment. So we put a plant in control temperature and a plant in high temperature, LTMH, and we looked at the pollen of different parts of that plant. That was part of the plant that is in its own temperature, but also we put one branch into the other room in each case, and we looked at the flowers on that branch. So if we first look at the plant that is in control temperature, it has nice high pollen viability, so 85% of the pollen is alive and well, whereas the pollen from the same plant, from the branch that was in the high temperature, chamber, there was only 1% of the pollen was still alive. And we did the other way. We looked also the other way around. So if we look at that plant that was completely in a high temperature condition, indeed the pollen from the flowers was very low, only 3% in this case was measured. Whereas the branch that was transferred into the control temperature, the flowers had very high pollen viability. So we repeated this experiment also even more precise with little bags, so we don't air bags around this around such a truss through which we flow cool air, and we get exactly the same picture. And the conclusion is long-term mild heat affects the flowers directly, and it is not an effect on the vegetative part of the plant. And we also measure photosynthesis, and it's indeed not affected by our type of heat. So altogether, we think it's really a local effect in the flower. That was our first basic question that we wanted to have answered. And then the second question is, well, when is the damage actually done? Because we're looking at open flowers, mature flowers, and we see these dead pollen. So, but pollen take a while to develop, so they take about 10 to 14 days in tomato. So where during that trajectory is the heat doing its job of damaging the development, or do we actually need heat during the whole 10 days? So we did this experiment in which we followed all the open flowers on a plant starting from the day that the plant was put in long-term mild heat. So this flower actually only had, this was an open flower on day 1, it only had high temperature for 24 hours preceding the day, and there was no problem. So actually, if you look at it roughly, there's not a big problem until even 8 days. So if you open a flower that has been in the heat, a mature flower that has been in the heat for 8 days, it's still pretty well in terms of pollen viability. Whereas if you then look at flowers that have been in the heat for 9, 10, 11 and more days, there they have big problems. So this was quite a strong LTMH in this case, LTMH effect. So we get below 10% pollen viability. So something is happening. At least you need to heat 9 days before the flower opens. And the other way around, we put the plant back into the control temperature, so out of the heat. And it took about 11 days. Here, if you look at that, it took about 11 days before we saw a recovery. So something is happening between 9 and 11 days before flower opening. So we conclude that the main effect is at meiosis and early microspores. We also looked at that under the microscope, and this is the stage that these flowers have at that time. And we found out that if you only give 4 days of the heat wave-like profile, that is actually sufficient. So you give 4 days of heat, you put the plant back in controlled temperature, and you wait 10 days, and then you see that the pollen are bad. Okay, so it is really only it's really a specific sensitivity around this phase, and afterwards it doesn't matter. The plants, you can take them out of the heat, and still 10 days later, a week to 10 days later, the pollen will be bad because they suffered at this stage. Okay, so something is happening in that stage, but do we already see that back in the pollen then already? Are they just dying at this stage? And that would make no sense, because we see dead mature pollen. So if they would die very early, why can we find them back in the mature flower? So we looked at this in detail under the microscope, and we put plants either in for 4 days in control or 4 days in the heat, and then we took them out, and that was all before this tetra stage. So yeah, so ending at the meiosis stage, and then we followed them, and we know that after if the flowers are open here, that would be 10 days later. These pollen are good, they come from control, whereas these pollen are bad. So we asked the question, when do they die? Between the meiosis and maturity. If we looked at tetrad stage flowers, actually we did not see any difference. So these plants had already been in this 4 days in the heat, but still tetrads were fine, microspores were fine, and we first saw the pollen dying after mitosis. So we have bicellular pollen, and here for the first time we see a difference. Okay, so we have an effect at meiosis, early microspore, but we see the effect much later after mitosis. Okay, so this is our answer to when is the damage done. Good, so for a long time, so there's actually, if you look at literature, many people noted that if you look at these mature pollen from a heat treatment, they usually have reduced starch accumulation. In tomato, you would have to look a little bit before maturity, because really at flower opening, the starch is actually already converted back to smaller molecules, carbohydrate. But just before, we see exactly the same thing. We see that pollen from the heat, they have much fewer starch grains. For a long time, we thought, well, that's nice that everybody sees that, and you can actually use it in some species. It's used as a staining to look at the viability. But this must be the result of pollen dying, because this effect was so much earlier. We affected meiosis, microspores, and well, if you then end up with fewer starch granules in your pollen, that must be just a late effect of the result of dying pollen, and cannot be the cause. So, but now we've seen that actually the pollen indeed die very late. So there is actually a bigger time window in which maybe carbohydrates metabolism might play a role. So we decided still to look at this. So although this is a very late effect compared to when the heat does its job, we think it could be something that maybe the heat does affect carbohydrates, and that's the cause of pollen dying. And we looked at this by looking at the sucrose and the hexoses that are made from sucrose, and there looked at their levels in anthers, either in microspore stage, this is late microspore stage, or in this bicellular stage where we know that the pollen start to die. And if you look at sucrose, actually there's no problem. The red line would be the level in the heat treatment, and indeed these anthers have nice levels of sucrose. But if you look at the hexoses that are made from this sucrose, we see that they're always lower already earlier. So not only at bicellular pollen when the starch is being made and not enough of it is made, but even earlier. So indeed, we think, well, maybe actually we have a problem here in hydrolyzing sucrose to the hexoses for the pollen, done by an enzyme called invertase. Maybe there's a problem with reduced invertase, but we're still not convinced, because this is just a correlation and we would like to complement it. So that would be actually the ultimate test, to prove that, for example, low hexose levels are really upstream of pollen failure, so they are the cause of pollen failure. You would like to complement it by giving extra glucose, extra to the plant. And that has been difficult. We've tried it a few times, but it's very difficult to complement that specifically. So we're still thinking about that. We would like to do overexpression of invertase, have not come to it yet. Knockout invertase inhibitors, so those are proteins that actually block the activity, or treat with chemicals that improve this energy metabolism. But we're still on this. Okay, another example of a physiological process that we studied whether maybe hormones are affected. Because, well, many hormones have been implicated or are necessary for good pollen development. So we thought maybe something is happening in the balances or the levels of these hormones, and that is actually leading to a problem with pollen. So in this, I will just give you a small example. So here we measured jasmonic acid, and we see that in the heat, indeed, the jasmonic acid goes down. So this is a hormone which is known to be necessary for male development, and it goes down in the heat. Okay, so jasmonic acid levels are lower. So we asked the question, well, what happens then if you have a genotype which already has quite low levels? Okay, so that genotype was available, and it's called DEF1 mutant. It has low GA levels. But still, if you look at the control temperature here in the bar, actually it can make pollen quite nicely. But if you put it in high temperature, it's very sensitive. So this was a very mild high temperature. Our wild type was hardly affected here, or not affected by the high temperature. So it was not too high. I think we used 30 degrees in this case, but already this genotype has big problems now to produce good pollen. Well, we would also like to complement it to see, well, what if we add actually extra jasmonic acid? And we've not been able to get that working. It's really difficult to apply chemicals to flowers and not actually get negative effects or get effects in your control. So instead, we thought, well, maybe there are genotypes with higher jasmonic acid signals or levels, and we came up with this idea because it's well known from the defense field that jasmonic acid and salicylic acid are let's say, working against each other. So if you have salicylic acid, it dampens the sensitivity to gismonic acid. So we said, well, maybe if we have a plant with low salicylic acid levels, it is more sensitive to gismonic acid, and maybe that does something with the pollen viability. And indeed, we also have this genotype available. It's a transgenic plant. It has very low salicylic acid levels. We've measured it, it's true. And this plant is hypertolerant. So in this treatment, we had a bit higher treatment. Wild type is nicely affected by the heat. And this line with lower salicylic acid levels was hypertolerant, better pollen performance. So indeed, we think that low gismonic acid might be a cause for pollen failure under high temperature. But still, we would like to complement. So we're still working on whether we can not do it via changing salicylic acid levels, but either genetically or with a treatment apply more gismonic acid and see if that helps. That would be a nice test. At the same time, we've generated a hypertolerant genotype, or identified a hypertolerant genotype. That's an example of where we go from question 1 interacting with question 2. So now, because we understood something about question 1, we actually end up with a genotype which is more tolerant, answering question number 2. Good. So we looked at more physiology. We looked at ROS levels, had some nice results there. But I would like to skip those things and answer one last question about the physiology with you. And that is a question that has been bothering me for a long time. I've actually introduced it to you slightly, and that is long-term mild heat actually what is happening in a heat wave, or is a heat shock? So that would be a very sudden increase to a high temperature, which is also a heat stress. Would that be happening in the field? Well, first, to answer that, we said, well, are those two things actually the same? Is the stress that a plant perceives from a long-term mild heat the same as the stress it perceives from such heat shock, which has been studied very well because it's much easier to study, of course. You can just give a plant a few hours of heat and that's your experiment. Okay, so are these the same? And we got some interesting results to answer this question. I would just go to the overview with you. So we had plants which we knew, we had tested ourselves, were tolerant to long-term mild heat, and we also had genotypes that were affected in their heat shock tolerance. But this also affects pollen. We've measured it, you can nicely see it. So we had some plants that were affected here. I'll go through it with you. So we have this low salicylic acid line that I just introduced to you. It has high tolerance to long-term mild heat, so the pollen do better than the wild. We have another genotype. I'll come back to you later with that. It's called Narcalang. It's an old landrace, and also we know and we've measured that it is tolerant to long-term mild heat. So what we did is put these same plants in a heat shock, and interestingly, those plants were not better when you put them in a heat shock than the wild types. Okay, so this does not lead to also tolerance to high heat shock conditions. And we also did the other way around experiment. We had a line which had an overexpression of heat shock factor 2, and we saw that it was very tolerant to heat shock. But if you put it in the long-term mild heat, actually it's not doing better than the wild. And here we have a line which has low heat shock protein 100. It's an important chaperone under heat shock. And indeed, if you put this plant in heat shock, it is not doing very well. It has problems. But if we put it in long-term mild heat, it does perfectly well. Okay, so altogether, we can already say here that long-term mild heat and heat shock are different stresses, and they need different tolerance traits. So looking at all the literature on heat shock, it will give you much information, but not about what is happening under long-term mild heat. It still doesn't answer what is happening in the field. We'll come back to that. Which of these two is actually more relevant in field heat waves? Good. So now I would like to take you to question number 2. That is the existence of heat tolerance. And we've not, I must say, we have not worked much on treatments that change heat tolerance. So there is some literature, certain compounds, if you put them on the plants, that they seem to do better. So that might be something that we'll work on later. But for the moment, we've looked at genetically induced heat tolerance, whether that exists. Good. So to have a look at genetics, of course, we need to have some phenotypic diversity generated from genotypic diversity. So we did this experiments quite a few years ago already, and we looked at a lot of wild tomato related species, and also looked at cultivars and land races, and we screened them for tolerance of the pollen development to long-term mild heat. So here you see, just to show you the range of tolerance, we have lines that were in our treatment did not produce any viable pollen anymore until lines that still produce up to almost 40% viable pollen. And that is quite a lot. 40% would actually be sufficient to get almost full seed set. So a broad range of phenotypic variation. And because for these wild accessions, we knew exactly where they came from, we did a small additional experiment, and we asked the question, well, if we look at the average annual temperature that these accessions came from, does that have any correlation with how well they can stand high temperature? And we see, and you will appreciate that with me, there is a positive correlation. So plants that are collected from places that are on average have a higher temperature, they tend to do a little bit better in terms of heat tolerance of pollen viability, but it only explains a little bit. So there's a large variation still around this factor of mean annual temperature. It might help, it might predict a little bit, but not everything. But something else we noticed in this screen is already that lines that were known from literature to be heat tolerant in the field. So especially already in the last century, there's quite a lot of literature from big field experiments, field trials, and some genotypes popped up as heat tolerant, and they've also performed well in our long-term mild heat controlled conditions in a climate. So that already starts to answer whether LTMH is a good proxy for the heat waves, or whether we should have been looking at heat shock. Good, so from these screens, we found that this is actually an example of something which was already known. It's a Philippine landrace Nagkalang. So it's been reported before by others to be very nicely heat tolerant in the reproductive phase. And we found exactly the same. We did in this case a simple biparental mapping experiment to find out what the basis of this tolerance was. So here you see the QTL plot for three traits: pollen viability, pollen number, and style protrusion of this biparental population. And we identified a single QTL which explains everything, the whole phenotype, or the whole diversity of pollen tolerance that we saw in the F2 population. So that's very nice. If we look in the F3 and we look a little bit better at this QTL, we see that you need it in a homozygous state, the Nagkalang allele, to get this higher pollen viability. And so it gives about 25% higher pollen viability, but it's a recessive trait. You need it homozygous. And that also tells us that this locus is doing something in the sporophyte and not in the pollen itself, because if it would be in the pollen, the heterozygous, half of the pollen should be protected already, but it's not the case. We don't see a sign of that. You need it homozygous, so probably it's doing something around the pollen in the flower, we think, because it is a local effect of the heat. Maybe in the tapetum, that is a cell layer that is just around the pollen. Okay, we have a few conclusions from this experiment. So we identified the first QTLs for male heat tolerance. So I think actually for all plants, we did it it's the first one in tomatoes. We found it from cultivated tomatoes in Nagkalang. We also found some from wild tomatoes, which I haven't shown here. But actually there's also very little information from other plant species. So this might well be the first QTLs for male heat tolerance altogether in plants. Then we see that actually if you look at these individual reproductive heat tolerance traits, like pollen viability, pollen number, we are not looking at a polygenic trait, maybe a multigenic trait in some cases, but some cases even monogenic. So it's not a very complex genetically if you separate these traits from each other. If you would look at that as fruit set as a whole, of course, all these QTLs will have an effect, and then it becomes polygenic, I would say. Well, I can't say too much, or I can't say anything about it yet, but I have a PhD student who has fine mapped QPV11 is what we call this one. So that will give much better markers. So we really have close by markers to follow this trait and to actually use it also in breeding programs. At the same time, we hoped from the identity of the gene that is affected, we hope to take that back to question 1 again. Because if we know the identity of this gene, we can start to ask, well, how could this gene with its identity, with its activity of the protein, how does that lead to higher pollen thermotolerance, heat tolerance? Okay, so there is an example of things going back to answer the other question about the mechanism. Good, so then to conclude, I would like to say a few words about a very recent experiment that we're doing. I already told you we're now taking things from our greenhouse and climate rooms, but also into the field. So this is a big experiment where we made a 4-parental rail. And we took it into the F6 generation, so it's nicely, largely homozygous. We have 400 lines, and we phenotyped it in our chamber in long-term mild heat, but at the same time, we phenotyped this population in the field. So it works only once at this. We put the plants in Italy and Spain, and there's variation, as I said before. In Italy, we did not encounter a heat wave this year, last year, but in Spain we had a very nice heat wave over our plants. So this was a field in which we had 400 lines in 3 complete and randomized blocks, and each time we had 10 individuals per line per block. So this comes to 12,000 plants were in those fields, and they were phenotyped by us in detail for pollen viability, pollen number, things like this, pollen release. At the same time, they were phenotyped or evaluated by the breeders at the end of the season to estimate yields from all the different individual plots that we had. Okay, so this is something we have to look at carefully, but it was just that it made you, and actually it made me realize that there's of course a lot of statistics involved in these kinds of experiments, because your field is not completely homogeneous. You might have more wind at one end, a little bit more water at the other hand, and so that might all influence your results. So this is the raw data. So if you have this raw data, because we had all these replicates in here, we could find out a spatial trend. So there is something happening here in the edges, which makes the plants behave, whether separate from the genotype, it makes the plants behave different from the ones here in the middle. So that was all corrected for, so we get this fitted data. And if you use this fitted data, look at the genotypic effects on those fitted data, there's almost nothing else left. So actually, here is the fitted data really can nicely be used to look at genotype effects. Okay, so we have taken out most of the environmental differences in those fields. So this is all statistics that we've been doing with a dedicated statistics group from Wageningen University. So what did we learn from the field experiment? So this was already interesting to show you. We looked at our pollen viability estimates that was during the heat wave, or actually it was a little bit after the heat wave started. We took into account, of course, those 10 days that you need to affect the pollen. So when there was the heat wave started, a week later, my PhD student, with some help, they traveled to this field, they measured pollen viability. And as I said, at the end of the season, fruit set was estimated from the same plots. And although the heat wave was only part of the whole growing season, already we see a positive correlation between pollen viability and fruit set. Okay, so higher fruit set was better plots according to the breeder, and well, this is the pollen viability from bad to high. It's transformed for statistical purposes. Okay, so although the heat wave is not affecting all the fruit set in these fields because it was just there for a few weeks, still we already see this positive trend. So that shows that pollen viability is also in the field. It shows us at least that pollen viability also in the field is important for fruit set and for yield, because that's what we all want at the end, of course. And we predict that if you would look more specifically at fruit sets of the flowers that were really in the heat wave, so those exact flowers that we know the pollen viability was good or bad in the heat wave, then you will get an even much better positive correlation. And we'll do that in coming years. So this is just a picture, nothing specific on that, just to show you that we did find QTLs already, because all the rails were genotyped, so we did find pollen viability, pollen number, style partition, and even fruit set QTLs from this big field experiment in long-term mild heat. So the conclusion, QTLs can be identified in the field if you do careful phenotyping. We even found something for fruit set unexpectedly. But very important answer to one of our questions, that we find exactly the same QTLs that we found in our own climate room. Okay, so that does tell us that what we've been doing with our climate room is actually relevant. Good. So with this, I would like to come to some conclusions. They should sound familiar to you by now. Pollen viability is a central trait in reproductive heat tolerance. We've shown that it does correlate with fruit set in our experiments and even in the field late in the last experiment. Long-term mild heat affects the flowers directly, so it's not an indirect effect on the vegetative part of the plant. It's the flower itself that perceives the heat. Then we've seen that early pollen development is the sensitive phase, but the pollen die very late. So there's something happening in that development still. We're looking at that, what's happening between feeling the heat and actually dying. That's an interesting phase, of course. We see that there might be problems with sugar metabolism, has been hypothesized for a long time already, and we have now added the jasmonic acid signal as maybe being a cause for pollen failure under high temperature. So we're working on that. I've shown you that long-term mild heat and heat shock are actually different stresses. So they're different traits that give you tolerance to either of these. And we think that this one is a very important one for field experiments. I've shown you that natural variation is present, and it's actually, if you look at specific subtraits of yield, in this case pollen number, pollen viability, things like that, you can find very distinct QTLs. So these are mono or multigenic and not polygenic anymore. And I've shown you that performance in long-term mild heat is predictive for field performance. Okay, so we think if you would like to have germplasm which is tolerant to heat waves, we should keep working on this trait and might not put as much focus on the heat shock tolerance. Although it does exist, although pollen are affected by heat shock, this is not the same, and this is not what is happening in the field. Heat shock is when the plant goes all of a sudden from its optimum temperature, 20-25, you put it all of a sudden at 40 degrees for a few hours. It's a big stress, it affects pollen, but it's not representative of what's happening. Okay, so with that, well, we can quickly go through this. This is for the future. I hope to make a model out of all the, let's say, the physiological and genetic data that we're generating. So this is a long-term goal. I hope we can actually, let's say, make good estimates for all the different traits and QTLs that we're finding for how that affects yield in the field. And I think this the work that we do is also transferable to other species. We've started doing that, but also other stresses, because some other stresses, depending on the species, for example, cold or salinity or drought, might also affect pollen viability. So I think that there should be able to compare those stresses with heat stress, with the things that we're finding out. And one stress maybe I should mention in particular, because that is something I learned at the last APSA meeting that I already presented to you, and that is by talking to breeders that there is also a problem with air humidity, or relative humidity, because there are seasons, and I think that is especially in Asia, you have these hot seasons that can be either dry or hot locations that have dry heat versus locations or seasons where you have humid heat. And these seem to pose, and that is actually something I got from you last time, these seem to be two different types of stress, and you need different tolerance traits, different genotypes to be able to deal with the two different types of heat, so a dry heat versus a humid heat. So we're working on that as well, and we have started doing some genetics and seeing if we can find specific traits for dry and humid heat reproductive tolerance. Okay, with that, I would like to thank some people. So the people from my university, of course, that are helping here, some of them here in this middle row, Nikki, Jimeng, Hanjing and Florian, they've all graduated already and they are working either at breeding companies or at institutes. These are my current PhD students doing the work. And I've also been getting help from people outside of my own university. And I would like to say, especially Fred van Ewijk and Caroline de Kovel, who are from the statistics department at Wageningen. So they are a big help when analyzing field trials. So the funding comes either from agencies or the government, that's at the top here. And also I've had support from many different breeding companies already that have recognized that this is an important trait to work on. I'm grateful for that support. So with that, I can probably stop the share.
Thank you very much, Ivo. Very interesting. And I think
a little bit over time. Sorry for that.
No, but I think your presentation was very good, and I think a lot has happened in the last couple of years compared to your last presentation. When was that again in Bangkok? 2000?
I think it was 2017, probably.
Yeah, a lot has happened.
4 years ago.
Yeah, we have time for a couple of questions. May, how do you want to do that? They're in the chat box.
Yes, we received the questions from the Q&A box, and basically I think we have time to answer 3 questions because we have 10 minutes left.
So the idea is that I start at the top?
Yeah.
No,
yes.
Yeah.
We need to.
Yeah, so I just
yes,
start?
yes, please.
Okay.
Yes, you can.
So
yeah.
the first one, what are the effects of long-term mild heat and heat shock on ovule of ovary? In hybrid seed production, pollen can be supplied from region of normal. Right, yes, I understand your point. So your pollen can be quite optimal in this case. You supply it from another region, but your female plants are still exposed to the ambient conditions. So that indeed, I did not mention that there also is an effect on the female side. So I think if you look at literature and also a little bit what we found, we did a few experiments, not a lot on that, with reciprocal crosses, actually exactly what you're describing here. And we do see effects on this female side, but they are less severe. And that's also what you find in literature. They are less severe than on the male side. So whereas you, for example, you might drop a fruit set, let's say, or seed set till 20% if the pollen were in a certain heat condition. If you, from the same heat condition, you look at the female side, you might still have 50% fertility. Okay, so indeed it does affect, and in your case, it is in this case, I agree in that the female side is the main factor still. So we have not looked at it, but it certainly warrants some attention in the future.
I see similar question. So a similar question, but to hot peppers. Are the effects and causes explained in tomato for LTMH and HS is similar in hot peppers?
In hot peppers, yeah, I think yes, indeed. I think you have the same effect in hot peppers. Well, you can go even to monocots and you still see this same effect on male fertility. On female fertility, I must say I'm not an expert. So for that I would have to look in literature. I'm pretty sure that also female, so my guess will be that also female fertility, but certainly male fertility is affected by high temperature in pepper as well. So maybe I answer this. Another question, are there any other traits other than pollen viability we can observe phenotypically in the varieties tolerant to heat stress, especially in plant growth, leaf structure? Yeah, that is of course an interesting question. That would give you some markers, much easier to score markers for heat tolerance. We've done a few experiments that are mostly on seedlings, whether we could associate some vegetative traits to the reproductive performance of the same genotype, and we did not find it. Okay, so that fits to our idea that heat affects the flowers directly, and that it's really something different from what happens in the vegetative part. So one association that we did find, we've not worked it out, but I can still mention it, I think, is that something that breeders and farmers tend to call antheridial cone splitting. So that is where the anthers become wider, they become greenish. So they might tend to bulge out a little bit. So antheridial cone splitting, there might be a correlation with male fertility. Although I have to be a little bit cautious, I would not start using it as a marker, but straight away. But we've seen some associations there. So, please explain heat stress affecting vegetative growth with low photosynthesis, ultimately affecting yield levels and poor fruit quality. Yes, you're completely right. That comes back to the fact that, of course, yield integrates whole physiology. So indeed, if you have temperature affecting the vegetative phase strongly, then automatically you will have an effect on yield in the end. So that is actually it's a different type of stress and different effects than what we're looking at. So we're looking really at the process of fertilization, really the reproduction itself. Whereas, of course, if you have much less biomass due to the heat during vegetative growth, you will end up with fewer fruits in the end, and your yield will be affected equally. So I'm not saying that our research is targeting the only thing which is important, and yes, so there are other things aside from reproductive heat problem. Photosynthetes, maybe to answer that, maybe to come back, the low photosynthetes are not the cause for reproductive failure. Okay, so we've shown that. There's the vegetative phase does a vegetative response does not affect, is not the cause for the pollen response to it. That is per flower, but overall you can get lower levels of flowers and fruits by affecting the vegetative phase.
Yeah, maybe the final question, Peter Hansen's question, in heat stress field trials.
Okay, in heat stress field trials, how many flowers? Oh, sorry, the question disappears now. Oh yeah, it's on the top.
I move up, yeah.
Yeah, they move. How many flowers per plot do you sample for pollen viability analysis? Oh, so to be honest, to give you the exact answer, I would have to ask my PhD student. So we obviously run through these experiments before, but how that turned out exactly, I'm not sure. But it was they sampled over multiple days. So we never work with one flower per plot or anything. So we have been sampling from the middle of the plots. I remember that because these are ground determinate variety. They are growing on the soil, so they're not nicely separatable in the field. Well, you know all about this, of course. So to be sure that we are actually sampling the right genotype, we've also always been sampling from the middle of all of those 10 plants in a row, and over multiple days. So my estimate would be that we have 10 flowers at least per plot.
Yeah. And then maybe Ivo, in relation to this, how do you have that the flow cytometer there on the spot, or do you? Yes. You analyze the sample on the spot, right?
Yes, we do both. We analyzed. Well, we don't want to store the pollen. We know that affects viability, especially if they have already been stressed. That would add up to each other. So we want to analyze them straight away. So we had the emphasis, actually multiple of them. We took them to the fields indeed to measure pollen viability. What we did do, so we vibrated flowers to get pollen out. We did take the rest of the flower, dried it, and took it home. And then during the year, we have been looking at the number of pollen that were still in all of these flowers. Of course, they were all dead. So there was not any more to look at viability, but to have a good estimate of the total number of flowers. And then we can actually say how many flowers were released by our vibration. Because that's where we can also get that trait. And the difference there. Some flowers are very bad in releasing pollen of some genotypes, and others are very good at it. and also in relation to the humidity and at that time point. Yeah.
Okay, I think we have to leave it to this. We will reply all questions and share that by email a few days after this session.
Yes, that will be my pleasure.
Thank you very much, Ivo. You're welcome to stay and listen to the second speaker. The second speaker, Professor Sheshshayee M from University of Agricultural Sciences in Bangalore. I was exposed to this topic of water use efficiency, I cannot remember exactly, but maybe 10 years ago, by one of Dr. Sheshshayee's colleagues, giving a talk for East-West colleagues. I was very much impressed and impressed by the technology that is used to measure water use efficiency. So I'm curious on what is the state of the art and your results. So please, the floor is to you.
Thank you very much, Professor Simon. Can I share the screen now?
Yes, please go ahead.
Yes. So you can see that?
Yes, we can see.
Yeah.
Maybe you have to put it in presentation mode. Yeah.
I did. Yeah. Thank you. Okay. So thank you very much for APSA for having invited me to talk about the kind of science that we are trying to do here now. As Simon said, that he must have listened to my professor, basically some of the information that he has given at that time. I hope I will not repeat the same thing, but we'll go ahead, as he said, what are the kind of state of the art that we have now. Actually, my life is much more easy now, thanks to Ivo for having given a lovely talk before and telling what are the problems that the plants have. And one of the things that we also see is something which is associated with the spikelets. But unfortunately, I should say upright that I have not worked much with the tomatoes and your cucurbits as such. In terms of vegetables, though we started some experiments now with the World Veg very recently, but I will not be able to share the data because I have not shared the data with them as yet, as of now. But what we are trying to do is if we can improve productivity under water limiting condition is something that we have. I mean, one thing that I don't want to get into the details about these kind of things is human being settled to do agriculture, and some people important people have believed that the agriculture actually is a disaster for the society, for the humankind, because of all the pollution that it causes and so on and so forth. But the human being settled to agriculture because I think agriculture would have been, at that time, was one of the much better sources of generating food for him and his family. And that's the reason why he must have shifted and even the animals sort of disappeared. But then if you very carefully see all the flourishing civilizations that are known, whether it's the Egyptian or the Mesopotamian or the Indian or the Indus Valley civilizations, all of them started in this where the source of water was plentifully available. Actually, it was the civilization or the civilized lives, or so-called, is strongly associated with water, is something that we always know. And these river valleys must have perished because the water availability decreased, which we know was the case in Mohenjo-daro and Harappan civilizations. Now, what we're trying to do is when you now have these civilizations disappearing because of water, and what may perhaps happen to us. Now, this particular graph, if you see here, is something that is happening, which is coinciding with that of the Green Revolution in the 1950s and 60s. There was a burst of the human population indicating that food was available, and then we could perhaps afford to have many children in the family. So the human population must have increased. Maybe, I mean, I was actually feeling sad when I saw this. This is when I was born, around this time when the growth rate was maximum. And then the question that we have is, as scientists from agriculture, do we have, can we stay secure in terms of food and nutrition, is a question that we always have. But when you look at the situation that we have in the world, and definitely in India, that the food grain production is actually coming down a lot. And then this, that what we have now, I mean, what we have now is something which is what we have now, in the sense that the annual growth rates of the cereal production is 0.6. This is wrong. This is not 2030. This is now. and what is predicted or what needs to be achieved in the next 30 years, or maybe next at least 10, 15 years, is about 3 times more in all this kind of crops that we have, cereals or the oil seed and the pulses, and definitely for vegetables and any of these kind of things. But if you see this graph very carefully, this was about 8 or 9 years ago, it was in Nature, that they show that there is a plateauing of the yield in most of the crops, especially the major crop that we eat like cereals, though there is some kind of a straight line in maize. Most of it is going for animal feed here. But then if you look at the cereal that humans consume, wheat as well as rice, there is a very strong plateauing in the last 10 to 15 years, perhaps. And even in India, this is from the Ministry of Agriculture and Government of India. From the 2013, there's not been a substantial increase in productivity at all. I mean, this is something which has to be bothering, which is bothering all of us for sure. And I'm also, I can sort of analogically tell that the kind of productivity that is increasing in vegetables is also sort of not very encouraging in terms of improving it further. But what we have is this particular yield plateau is coming because of resource limitation. And that is one of the major causes that is actually responsible for the kind of civilization that what we have. I mean, in India, for sure, it is, people believe that if there is if there's going to be a war next, it will be for water and not for any territorial other things. But this particular requirement of water has increased the withdrawal of water from the ground table. Groundwater, and the water table is depleting, and that can have a very catastrophic effect. Now, like for example, if you see this from the past 20 years or 30 years, there has been a steady increase in the amount of water that is taken out, and most of it is going to irrigation. And now, with the kind of a small rise that you can see here for industry and domestic purposes, the amount of water that is actually being used is stagnating again. And that is, I think, is going to be a severe problem, because that's going to cause a lot of gap between the population growth and the food that is being produced by them. And this is the kind of line that goes on straight to the sky in terms of the groundwater withdrawal. And this is the worst scenario in India, and especially, I mean, this particular kind of thing goes on for both, the food crops, food grains, as well as vegetables. And this is, in most of the places, it is for actually the growing of tomatoes and things like that. And there was a very elegant review that came this year in the month of February or so in Science Advances. And what they have done is they have analyzed the scenario of how water is actually being depleted from the ground. And you can see here the amount of the area under irrigation in India is not really changing for the tank irrigated and even for the canal irrigated things. But what is increasing is the well irrigated thing, and most of them are the tube well irrigated mechanisms. For the past, I think, 20 years or so, there's been not a significant change in the average rainfall over the country, though there are some kind of deviations, and we call that anomalies in atmospheric sciences. But then what is increasing significantly is the withdrawal of water from the ground or from the water table. So the groundwater is disappearing. And they had a very nice analysis to sort of compare and tell what would be the kind of probability that the people would actually go for getting the water for irrigation from bore wells or the tube wells when compared to any of the other sources. You can see that the probability is deviating away from that, suggesting that they prefer to use the wells and then the tube wells and groundwater for irrigation. And even the persistent of the crop area is also sort of away from the tube well, suggesting that the tube well is actually the one way to go forward in agriculture. And what is going to happen in the next few years is something that they have done very beautifully. When in India, I'm sorry, I could have taken the world, but then we don't have the data. They have this Indian analysis that has been done. Intensely cultivated areas are somewhere here in these colors. But if you see now, with progression here by 2025, this is perhaps now, or maybe 10 years ago, by 2025, we're going to have some kind of severe problems. But in the most cultivated or intensely cultivated areas, you will have critically depleted levels of water in all these places. And this is where most of the agriculture is being done, and that's going to be the disaster. And by that time, by let us say 2030 or 2050, Indian population would be more than the Chinese. But there is some kind of a calculation I thought of doing to tell how much is the water that is actually available for humans to consume in Indian scenario. So in terms of the per capita availability of water, the availability of water is actually depleting very significantly, and what we have now is about 3,700 liters per person per year per day. I mean, this may look like very high because the urban planning suggests that 135 liters of water should be enough, and then in the rural areas it could be something like 70 or 65 to 75 liters of water per day per person. But this is the amount of water that we have available in it. But if you now calculate what is the amount of water that is required for a diet, which will now include both vegetables as well as the kind of other cereals and the grain crops that we eat. And if you have a typical recommended diet for an Indian kind of situation, which is a vegetarian or a non-vegetarian diet, you can see that the amount of water that is required for a person. I mean, this is from the data that we can calculate from the water that is required for growing a plant. And we can say that we need about 1,400 liters of water per day per person. And if you are a non-vegetarian, it's, God bless you, it's about twice that. It's about 3,400 liters of water that is required to make these things. You can see that the meat requires much, much more in terms of the water that is required for making these foods available. Now, how are we to now address this question is the challenge that we have. Now, we have to now produce for the populations. There is no doubt that we have to have them fed. And how is it that we can do this under the conditions of limiting water resources? Ivo was talking about the heat waves that come, but then the heat waves do come even in India. And then when there is drought in the soil or in the atmosphere in terms of the relative humidity, we call it the vapor pressure deficit. And in such kind of situations, I don't know whether we'll be able to produce for the kind of billions of people that we have on Earth. Therefore, what is it that we can do is to sort of try to do is to reduce the water footprint of our productivity. There's one elegant thing again that has been done it's an example that I can take from the Canadian research, that the water footprint that we talk about, the grain yield and the water footprint for grain yield can decrease significantly if you increase yield. This is something that is the only way that appears to be done. If I increase the yield, the water footprint, I mean, in terms of the amount of water that is used for making that grain decreases. But you can see here, after certain levels of grain yield, this curve is sort of flattening. And then I do not know whether we'll be able to sort of break this and then improve this further. And if you look at the nutritive values of these things in terms of the protein yield, water footprint, I mean, you can see that you already have something like 14 meter cube for every kilogram of protein that are produced, or about on an average of about 2,000 to 3,000 liters of water that is required for growing any of these crops that we have here. Now, yield, as I said, is not increasing appreciably. What needs to be done is the question. So we need to sort of make an better understanding about how the plants actually respond to drought. There's a small cartoon that we made to explain that when the plants are growing in the field, you have water in the soil and they take up the water. And if there is no facility to irrigate it, mostly dry land or rain-fed conditions, the plants take up water, they're happy, but then they lose the water, and then amount of water in the soil goes down. And if the roots are not capable of reaching them, the plants start showing the symptoms of wilting. And this is something that happens in nature, in the field of the farmer. Now, plants experience stress as the transpiration exceeds the amount of water that can be taken up by the plant, by the roots. And this results in the reduction of the turgor or the cellular water relations, and that affects growth in terms of decreasing the area or decreasing the metabolism or decreasing all of them. And one of the very, very significant things that happened, like what Ivo was also talking about, is reduction in the reproductive growth efficiency. Therefore, yield decreases, and this has been very well documented. Now, the question that we wanted to ask was, is it possible that we can sort of improve this? And when we look at the number of resources that the plants require, but water is something which is the central thing that is happening here. And when we look at this, the plants that we need to sort of design and develop should capture the water better, which is in terms of the uptake by roots. If I were to have deeper root system like what we had in this, if the roots were to go till here, perhaps the plant should have delayed its wilting for some more time. That is the concept that we have. And of course, we need to increase the use efficiency of the resources. And if I replace the resource with water, it is the water use efficiency which Sreeman was talking about. What we need here is better carbon assimilation by the chloroplast or by the photosynthetic machinery. And then we should have better root system is the concept that we're trying to talk about. Now, what we are trying to say is that the success of this particular mechanism of crop improvement or improving the productivity in water limiting conditions is possible only when we identify very specific traits, like what Ivo was also talking about a little while ago, in terms of looking into the specific traits that can improve the productivity. And what is more important is to sort of phenotype them and try to capture the genetic variability in such traits is important. Now, when we look at the reasons why the yield decreases, and this was done in cereals, as I said, I'm sorry, we don't have the data for the vegetables yet, as I can share it with you right now, but maybe with time we can do that next time. But what we have is, if you understand this, that the yield decreases, and in the 3 cultivars that we checked, and this is a recent publication that we had with one of my PhD students who also works as a sandwich student in Wageningen Institution with Shinyu and Dr. Paul Strook. And what she did was to sort of compare these good varieties. There's a lowland variety and an upland variety. In all of them, there's a decrease in the yield. But you can see that when there is a stress that happens during the during the flowering and then the grain filling stage, the reduction in yield is much more severe here, which indicates that in the GS2, there is a spikelet associated or spikelet fertility associated, mainly the pollen fertility or pollen viability kind of issues. And in the GS3, it is both spikelet associated as well as the grain filling mechanism. So it's a complex situation that we look at how the drought can affect the physiological processes. But we wanted to sort of make an analysis of putting all those kind of complicated things that are around here. If we want to now adapt to drought, if we want the plants to adapt to drought, we classified them into 3 mechanisms. We have the morphological traits, including the leaf area, leaf thickness, root morphology, and all those things. And then we said that we look at the cellular tolerance mechanisms, or we classify them as acquired tolerance mechanism, which are associated with osmoregulation or the reactive oxygen species scavenging mechanisms, protein stability, and so on. And then we have certain physiological traits. I mean, I can say that most of them are physiological here. Very specific physiological traits such as carbon assimilation, and then the amount of carbon that is flowing into the leaf. Now, what is the mesophyll ability to sort of transport this, and how good is that translocation in the phloem and so on, which will all lead to 2 things simply. One is whether the morphological traits are responsible for uptake of water, then you have water relations. And then if all the physiological things such as the acquired and the other things are responsible for metabolism, we are trying to now increase the carbon metabolism. So all mechanisms simply point towards maintenance of metabolism and water relations. So we try to do this. We wanted to suggest to the breeder, like instead of looking for selecting for yield under stress per se, why not we look at specific traits and try to improve them? And then the concept is that when we select for higher absolute yield under stress, there's no doubt that this is a very simple way, and then go put all the germplasm, pick up the one that has the highest yield. There's no doubt that the progress has been rapid, and that's what the breeders did. And if you look at it very carefully, there is very less variability among the already developed varieties which have higher yield under a drought, and then there's a high genotype and environment interactions. When compared to if we were to now have a very specific drought adaptive trait, there's no doubt that the progress can be slow, but if we have specific mechanisms by which we can phenotype them with a high throughput mechanism, and because of the low genotype to environment interaction among the traits that we look for, I'm very sure that the analytical method or a very specific physiological breeding can be done, is what we are saying. What we did, and from here I would like to show you some of the results that we have done for how we can perhaps do this kind of physiological breeding in cereals. And we know that the roots are one of the most important traits, and this is a very nice article that was written some time back. And then they showed a deep-rooted individual which has the near isogenic line for a particular QTL called the DRO1, and then that particular DRO is deep-rootedness in rice. And when they put it into a high-yielding water-loving IR64, it started becoming better in terms of that. So we wanted to do a similar kind of thing. So we analyzed a large number of ethyl methane sulfonate induced mutants in a particular variety called Nagina 22. And then we had developed a mechanism to now screen a large number of these mutants. We screened about 3,000 mutants in the stabilized mutants. And then for a root growth in terms of root structures, we have a tank which is sort of about 5 feet tall and made of bricks, and then we can dismantle the bricks and use a jet of water to clear the soil, and we can get the extracted. And what is the advantage in this case is the plants are grown in the same population that is actually grown in the field. So the inter-plant competition will be as you can see it in the field. Therefore, this phenotyping, a little back-breaking activity for sure, but then will give you a very, very specific very precise estimate of how the roots are growing. Then we analyze these root mutants. We developed mute map populations. We did both the genetic as well as the physiological analysis, and we showed that the high root genotypes had much higher biomass than the low root individuals. And then these guys had the deep-rooted plants always had much better photosynthesis under even underwater limited conditions. We stressed them to an extent of about 60% of the field capacity, and they had better water relations. And these guys had better spikelet fertility, and therefore they could maintain better yield. You know, the deep-rootedness, and there is no doubt that the deep-rootedness is important for taking up better water from the soils better. But there is a caution that we have. The moment you increase the root system, and in conditions when the water is very limited in the field, increasing the uptake of water will perhaps exhaust all the water that you have in the deeper layers, and therefore it's going to be something tough. So to that, we wanted to now superimpose on top of it how the plants can actually be efficient in using whatever water they can capture. So which means we are not sort of arresting them to go down and then take water. We're not stopping that, but then we say that you access the water, but then use it efficiently without sort of wasting that for biomass. And therefore, we wanted to have some mechanisms to use it for capturing the genetic variability in these traits. Phenotyping, of course, is the most important thing. And besides a number of things, what we developed was to use a technique. I'm glad that I did my postdoc with Graham Farquhar in Australian National University and Richard Richards and CSIRO, with whom I contact very regularly in terms of discussing about the stable isotopic application as a tool for understanding how to capture the genetic variability in physiological traits. You know, what you have is some of those important elements that are there in the periodic table that have biological relevance of these hydrogen, nitrogen, and oxygen and carbon. And when you look at the two kinds of them, these are all the stable isotopes. There's nothing to be the radioactive. And we remove the deuterium and the carbon-14. Thank God we do not have radioactivity for nitrogen and oxygen. I do not know what would have happened to the atmosphere if they had. But when you look at this, these isotopes have one extra neutron, and therefore they're heavy. But what happens is, because of this, there is a chance that either of these two elements can be taken up by photosynthesis or by any other physiological processes. But please be aware that the heavy isotope that you have is very, very scarce. And in terms of the natural abundance, it is very highly depleted in the nature. But it's something like 99% of all carbon is 12 carbon, and there's only 1% of them is 13 carbon, and so on. Now, so what is the question? Now, the composition of these, for example, if you take the ratio of 1 divided by 99, that's the kind of composition that we talk about, or it is 0.02 divided by 99.98, not 98, 99.98. I mean, when you look at this, it will be a very small, very, very vanishing small kind of a ratio, but that will give you quite a bit of information, like how I will tell you. The composition of these isotopes is very, very different in organic substances which are made by the process of biology. During these biochemical or even the diffusive processes, which we classify them as biophysical process, these isotopes, or the composition of these isotopes, are altered slightly. And if you can now identify and make measurements precisely on what is the extent of change or alteration that they had in the isotopic composition, we can very clear inferences we can draw on the process that was actually causing the kind of isotopic fractionation. We call it as isotopic fractionation. To cut a very long story short, we only did this kind of a measurement in a large number of crop species, and we show that the carbon isotope ratio, which is called the delta 13 carbon, is actually related to water use efficiency strongly. And therefore, being a high throughput mechanism, there we can use this technique very effectively. Australians have done this excellently, and Graham Farquhar and Richard Richards, who visited my lab, and we worked together. And this technology was used to develop a very good number of high yielding varieties under very water limiting conditions like Disdale and Rees. But there is always this problem, and this is a facility that we have in Bangalore. I mean, whenever it is possible, you're welcome to visit, and we can always make measurements on samples. It's a national facility which has to be accessed by anybody. And what happens is that when we now looked at high water use efficient genotypes, unfortunately, the breeding was never successful, because if the availability of water in the crop growing season is high by some rainfall or some irrigation, the advantage of your crop variety that you developed by breeding for high water use efficiency disappears. This is the major problem that we have for this. That is, high water use efficiency perform only under water limiting conditions, and others did not do well. But that is therefore not a very good physiological trait. So what we wanted to do was to now increase the water use efficiency without decreasing the transpiration. This is exactly where the difference was in terms of understanding the whole thing. So we needed another mechanism by which we can identify or to capture the genetic variability in these traits, so in transpiration. So we used a very similar analogy that people were using at that time in explaining how the water is evaporating from the ocean. And whenever water evaporates, it is the oxygen, which is lighter isotope, will escape faster. H2O16 will go away faster, which results in the enrichment of the oxygen isotope in the water that you have in the leaf. So transpiration is an evaporative process, therefore it should have definitely the same kind of an influence. And we measured the oxygen isotope ratios in the biomass, and we could develop that into a very beautiful technique to estimate the differences in the transpiration rate. And this is what we find, and whether it is in the controlled environment or the genetic variabilities, we find a very strong relationship between the transpiration rate and the oxygen isotope enrichment in the leaf biomass. This particular method has been used. Now we combine both the methods of using the oxygen and the carbon isotope, called the dual isotope approach, for identifying carbon fixing types, which we call the chloroplast or the mesophyll capacity types for increasing the biomass. And this particular method can be also extended to measure if the root system is better at all or not, because any genotype that has a better root system can go and access water deeper, and that will have a better transpiration, and therefore we can use the oxygen isotope enrichment is also sort of related to the root biomass in rice. So this was an excellent situation for us, where we had two proxies which were high throughput mechanisms to measure genetic variability in water use efficiency as well as root traits. And we wanted to now check if increasing the water use efficiency would generally improve the productivity. And in large germplasms of both rice and groundnut, we showed that the low root types will have higher water use efficiency because they are stressed under the field condition because of the low root system here. Therefore, at a given root or given leaf area, we showed that the carbon isotope is actually related to yield, which was not earlier not noticed, because we used all the kind of variability that we have in this. So when you now have a given leaf area or given root system, water use efficiency actually will be related to the total biomass and total yield. Therefore, a genotype with low or high water use efficiency will show high biomass is now possible, unlike what was previously known, why they said that the water use efficiency cannot be bred. Now what we did was to check if that is true if you tried to genetically. So what we identified was a germplasm line which had better root system called IAT 15960, just a number. And then there was a cultivar which was actually grown under puddled condition, and we crossed them, and then we developed a large number of recombinant inbred lines. We did the QTL mapping in it, and I'm not telling you the whole details about it. We did the QTL mapping. There's a biparental analysis, And then we developed these reels and then identified some of the individual reels which had the kind of introgression of the trait. So what I mean to say is Tanu had better water use efficiency and IET 15963 had better root biomass, with a significantly better root here in IET, and it has much better water use efficiency when measured using carbon isotope ratios. And what we did was we identified several trait introgressed lines where both the root as well as the water use efficiencies were high. For example, the trait introgression line had the similar water use efficiency like that of the donor line, that is Tanu, and then it also had the root system like how it was in the other genotype called 15960. So that means the trait introgression line is a deep-rooted, high water use efficient genotype, and lo and behold, it had much better stomatal conductance and photosynthesis when compared to the other two guys, and the yield was about 20% more. And this is something which is interesting. This particular plant is requirement of water, though it can transpire, but the translation in terms of the amount of biomass it can make for the same amount of water that they transpire, this had much better yield than the other genotype. And I was actually very happy. And you can see this is the parent one which had bad root and the parent two which had better root. This fellow had better water use efficiency. And now when you combine them, we had the advantage of both of them, and this variety has now been released for farmers to cultivate in the name Daksha. Daksha in Sanskrit, it means efficient. So we got this variety, and then we have a large number of them now. So what we did was we did an association analysis, and we identified the QTLs by population genetic approach for root and water use efficiency in a larger germplasm lines of rice. So because we had some success in the previous experiment that we did with two genotypes, we now wanted to introgress it from very different donor lines. So what we had was, we had a much better root genotype donor line from the germplasm, which is an accession called 39020. We crossed to IR64, and similarly we identified a better water use efficient, that is the low delta 13C time, and also crossed it with IR64. We combined the two F1s, and then we developed the double cross F1s. And this double cross F1s, now we did a marker assisted foreground selection for the kind of marker that we had. We had 18 markers linked with the traits that we had done by association mapping. And we then identified the ones that had the kind of introgression, and we back crossed it. So when you do the back crossing, we did 3 back crosses. We developed double cross, back cross 3 F1, and we had 140 of them. And we did, in all the cases, we did the foreground selection with 18 markers. And at this stage, at the BC3 F1 stage, we also did a background selection for identifying how much of the genome from the IR64 that we have recovered. By the time we came to this one, it was already about 88 or 92% of the IR64 genome had been recovered here. And then we allowed it to self, and then we developed BC3 F6 lines. And at this stage, they had almost about 98% of the IR64 genome recovered, plus the foreground marker that we had transferred for the root, as well as the water use efficiency into the same IR64. And what we did was to now measure a large number of traits, including spikelet fertility. This is where the kind of discussion that we had with Ivo, and what I am trying to say in terms of the yield maintenance under water limited condition is similar is the spikelet fertility. The spikelet fertility, you can see that there's no doubt in the water limited condition, there's a slight decrease, but you can see that the IR64 is very sensitive, whereas the trait introgression lines are not. And the roots were better. Because of that, it maintained better water relations. The relative water content of the tissue was better, and the carbon isotope ratios were better in the trait introgression lines, and the leaf area was comparable in similar situations. But the moment we put them to water limiting conditions, the trait introgression lines maintained better leaf area. Now, how is it possible that you can have so many different kind of things that are happening by transferring two traits? What I'm trying to say is, when we maintain the water relations and when we maintain the metabolism, I'm sure that the water relation associated leaf expansion is maintained here, And that's the reason why it maintains better leaf area, though the leaf areas are not different in the water, well-watered conditions. And because of this, because of the better spikelette fertility that we maintain in the trait introgression lines, and with the 98% in the background of this one, of the IR64, the trait introgression lines had much better grain yield under water limited conditions. This is something which is interesting. And therefore, that we believe that the trait introgression lines performed actually better under better than IR64. And then we analyzed these things for more close variations from about 260 of the trait introgression lines. We identified 26 best ones, and they had better water use efficiency because of the reduction in delta 13C. There's an inverse relationship between them. Higher grain yield they've had, and then we identified still 6 more lines which are actually now in the pipeline for the farm trials and other kind of things that are being done before we release the farmers cultivation. And you can see that in all the kind of stresses that we have, even at a very severe stress that we have, these trait introgression lines maintain better grain yield. This is something which is interesting, and this is a percent change over IR64. It had much better grain yield under severe stress also. And one thing is for certain at this stage is that when we now do these kind of analysis, I mean, kind of experiment that we did was to prove that introgression of these traits are possible. But one thing that is required at this stage is a very, very thorough phenotyping facility. Now, what we have here, if everything goes well, there's a small video for about 1.5 minutes after this. And it shows that we grow these plants in mini lysimeters. And these containers will have about 30 kgs of soil. And in this scenario setup, we did the tomato analysis as well. But what I'm trying to say is here, each of these things is automated. And we have these things kept on a balance. And the balance records the weight of the container in real time. And then when the water is being transpired by the plant canopy, and when the weight of the container decreases by about 50 grams, then there's an automated system which recognizes this and adds water to bring the soil to the same level. And this, I hope you can hear this. We developed a platform here, which we call a hybrid platform at university. Yeah, that's the facility that we have there, where the water used by the plant can be very accurately measured. And then we also have a non-invasive technique of capturing the images in real time so that we can measure the growth as well. So to conclude this, there is a possibility that we can now do the kind of physiological basis of drought adaptation or yield determination for crop improvement. This is for sure, it is possible. And phenotyping under natural environments, it's not the kind of controlled environment that we have. The plants are exposed to naturally varying conditions in terms of the diurnal and seasonal variation that may be there. And we record all those things and bring in kind of the corrections by the augmented mechanisms. And therefore, the phenotyping is now possible. And we have a large genetic resources these days with everybody. We have germplasm, we have mapping population, now we have mutants. And when you have all these kind of things, and then we can phenotype them accurately, and when we can now identify the markers and with the help of the genomics, I'm very, very sure that very strategic physiological breeding for crop improvement is definitely possible. And I should really thank APSA for putting us together, and I'm really happy that the seed industry, which is basically a breeder-driven program, called two physiologists to talk. I'm so happy about this. But before we end, I must thank the funding agencies. We've been receiving funds from support from the Department of Biotechnology. And in a very recent time, we had an Indo-Swiss collaboration for the genome analysis in a millet species called finger millet. And I should really thank all these young guys who have been working with me. Many of them have finished their MSCs and PhDs and have gone on. And then we have collaborations both in India, in number of institutes, but most of that is working basically in rice. And we also have some international collaborations, such as in Wageningen with, as I said, in the Wageningen University of Research and Agriculture University there, and then you have an ETH and Australian National University and ICSAT. Thank you very much for your patient hearing, and if there are any questions, I'll be very happy to address. Thank you so much. I can stop sharing now.
Thank you very much, Professor Sheshshayee. Very fascinating information you share, and it is. I do see that there is a chance that we can collaborate more, and I think that later on our R&D advisory committee will review this possibility and reach out to you, and we'll see what we can do for our APSA members. But really, thank you very much. You share very clear information about how the rules can help plant to use better resources, and that the end can increase their crop productivity. So now we have time for Q&A. I think 10 minutes, or we can take up to 15 minutes or so. Right now we received 2 questions.
Yeah. So there's the first question that we have is how would be the performance of these genotypes when they are selected under water scarce condition in sufficient times? Because water scarcity is that's what I said. This is what happens when you look at water limiting conditions. When you select for yield under water limited conditions, and then you picked up the one that are the best there, and then when the amount of water availability increases by whatever be the reason, it's uncertain, as he says very rightly, and if there is a rain or you irrigated it, then the advantage of your high yield decreases. That's the reason why we now believe that when you make selections for a trait, that trait should be responsible for protecting the yield under water limited condition, but it should not be a constraint for growth and productivity and when the water availability increases. That's the reason why we were much better off in terms of the results that we got when we combined water use efficiency with water use. That is, by combining the root traits with water use efficiency, we could, to a large extent, overcome this problem.
Right. Thank you very much. And that's
how the soil condition would help in increasing the increasing the micropores in the root zone that can hold more water. Okay. How can the soil conditioner would? Sure. Because, see, there are these. There are some of these compounds which people are using. There's a gel or some compound that people are using these days to increase the water holding capacity in the soil. I mean, these are management. For sure, there are a number of those kind of management practices that are being done. Mulching, for example, using plastic for mulch or something like that, and the degradable plastic that are available now, you can pour it back into the soil. All those things are being done. But then what would be much better for us would be is instead of adding something to the soil, some kind of a resin to the soil, why not we look for genetic enhancement? But no doubt that agronomy or management can also be one of the options for sure. Yeah.
Yeah, that's right.
What are the what is that per acre cost of
This
the system in US dollars? Oh, acha. What is the system? I don't understand.
Yeah, I wonder also, is that the image analysis or the...
Oh, the image analysis system or the whole platform that we have made? Is that what he is asking? I'm not sure.
Perhaps we can share through...
Sure. I mean, what would be best is that person can contact us. I'm sure I will be able to explain to that. And I mean, the plant eye is an internationally available. It is from the Phenospec's company. We've just bought it from them, and then we are doing the analysis. But if you want that phenotyping for water use, or the lysimetry kind of part, where we are measuring the water loss, and that is built by us in India. It was completely indigenous. And we can always work out on the cost of it in the US dollars, not at all a problem. I can discuss with him one-to-one.
Yes.
Could you
So,
please?
yeah, I think we can get his email and...
Yeah. What is that? Could you please share your thoughts possibly on combining root length and water use efficiency oh yeah, sure. That's what I said. In vegetables, for sure we can share, but not right now, unfortunately. But I can only tell you that the data that I have analyzed from what we did for the kind of variety that we had, but it's not a very large experiment in terms of germplasm. It was to compare between certain hybrids and certain high-yielding varieties. But what I can see is any genotype which has better root system and better water use efficiency definitely has higher biomass and growth rates and yield. This was in tomato. I can definitely share it after I share the entire data with World Veg. I hope that's okay.
All right. I think that would be interesting to learn actually.
Yeah.
Any more? I think that's all.
Yeah. No more questions. Good.
No more questions, right? Okay. So thank you very much, Professor Sheshshayee, for sharing very good concept of the of this technology. For I think that as I say, so according to what you have shared, also the research that currently has been done with Well Vegetable Center. After that, I think that would be interesting also to share more on the vegetables. And of course, we will reach back and find the way to collaborate more with you. So thank you so much.
Thank you very much. Thanks, May, and thank you, Mozam, and special thanks to Sitaram. Unfortunately, he couldn't come. It was he who was telling me that these kind of things, if you discussed, would be useful for the vegetable industry. I'm very happy to associate, and any sort of discussion we can have, there's no doubt about it. We can, Mozam has my telephone number if somebody wants to call in India or from anywhere, or we can always have the email for sure. Thank you so much for this opportunity, and I learned a lot as well.
Thank you so much. Thank you. And so we come to the end of the webinar. So first, I would like to thank our chair, Dr. Sreeman, and also our co-chair, who is Dr. Sitaram. Unfortunately, he cannot join in person on the webinar today. So I helped him to just do a closing remark. And also all the R&D advisory committees, I would like to thank for your input and suggestion for the webinar organization. Special thanks to our speaker, Dr. Ivo and Professor Sheshshayee. You have shared very good information. And for Dr. Ivo, there's some questions that we will reach out to you to get the answer, and then we will share to our participants. And for the attendees, and till the end of the webinar, we still have more than 100. We do really thank for your attention. And the PowerPoint presentation will be shared back to all the attendees by tomorrow. And this is very special. We start from this session that we will give back the certificate of attendance. But only one condition that you need to share us the feedback, because we really need your help to share with us how shall we improve the session and what kind of topics of interest that you would like to hear more from APSA R&D Advisory Committee. And so before we close, I just would like to share an information of our next ASCRT session. So this session will be organized on 16th of June. So, and the registration will be open early May. You will receive our communication soon. And the way to register is the same. So now I think you are quite familiar of how to do the registration. You just need to use username and password of voting representative, and then you just you can register as many as possible from your company. And so the next topic, we focus more on watermelon, and we will have Dr. Xingping Zhang and Dr. Srinivasa Rao present in the next session. So hope we will see all of you again in the next session. And today we would like to thank you everyone, including our secretariat team, for a good organization of this webinar. With that, I wish everyone to stay safe, and then we will see you all again in the next webinar on the 16th of June. Thank you. Bye-bye.